Cell signaling governs how our bodies maintain homeostasis, especially in glucose management. Every bite of food triggers a cascade of signals between organs, orchestrating insulin release and enabling cells to take up glucose. This mechanism is vital for producing ATP, the energy that fuels all bodily functions. In this blog, provided by Mindful Diabetes Inc. as part of the Pathways to Wellness series, we will explore this signaling process. We'll look at how food communicates with the brain to release insulin, triggering glucose uptake and energy generation. We will also examine how artificial sweeteners can interfere with this process, potentially leading to metabolic confusion and health concerns. Let's follow the pathway from the first taste of food to cellular energy production. Tongue-to-brain signaling, the gustatory gateway. How taste buds trigger brain responses. The process starts when food enters the mouth, and the taste buds detect sweetness, signaling the brain to prepare for glucose intake. Taste receptors on the tongue identify sweet flavors, sending signals via cranial nerves to the brainstem. The brainstem relays this information to the hypothalamus, which controls hunger and energy balance, preparing the body for glucose metabolism. The vagus nerve is also activated, linking the brain's signals to the digestive system, preparing the digestive tract for glucose. This signal informs the body that glucose-containing food is incoming. The brain signals the pancreas to prepare insulin release in anticipation of rising blood glucose levels. The digestive system begins to secrete enzymes needed for carbohydrate digestion. Cephalic phase insulin release occurs before glucose enters the bloodstream, as part of an anticipatory response. Brain to pancreas. The endocrine signaling link. Insulin's anticipatory release. Once glucose enters the bloodstream, the pancreas plays its critical role. The brain signals the pancreas's beta cells to release insulin, the hormone responsible for lowering blood sugar. Insulin is secreted into the bloodstream to regulate glucose through endocrine signaling. The importance of timing. Insulin release must be precisely timed. If insulin is released too early or too late, the body struggles to regulate blood sugar. Dysregulation can lead to insulin resistance, a precursor to metabolic diseases like type 2 diabetes. Insulin signaling and intracellular cascade. Insulin binding to receptors. Once insulin is in circulation, it binds to receptors on muscle, fat, and liver cells. Insulin receptors recognize insulin and initiate glucose uptake. This binding triggers a cascade of signals within the cell, starting with the activation of insulin receptor substrates. The intracellular signaling cascade. Insulin binding initiates a series of reactions inside the cell. Phosphatidylinositol-3 kinase is activated, leading to the activation of AKT. These signals help move GLUT4 vesicles to the cell surface, allowing glucose to enter the cell. Glucose uptake and ATP conversion, the cellular energy factory. The role of GLUT4. Once on the surface, GLUT4 transporters begin the critical task of bringing glucose into the cell. GLUT4 acts as a gateway for glucose to enter muscle and fat cells. The more insulin present, the more GLUT4 receptors move to the cell surface, increasing glucose uptake. From glucose to ATP. Once inside the cell, glucose undergoes several metabolic processes. Glycolysis breaks down glucose into pyruvate, which enters the mitochondria. In the mitochondria, glucose is converted into ATP via oxidative phosphorylation, providing energy for cellular functions. When this process goes awry, the impact of artificial sweeteners. How artificial sweeteners disrupt natural signaling. Artificial sweeteners, like aspartame or sucralose, can trick the body into thinking glucose is present. These substances bind to the same sweet receptors on the tongue, sending signals to the brain similar to those from real sugar. The body releases insulin in anticipation of glucose, but no glucose is delivered, causing metabolic confusion. The long-term effects on insulin sensitivity. Repeated false signaling can have adverse effects. Over time, the body's insulin response may become blunted, leading to insulin resistance. Without actual glucose to process, the body may store more fat, increasing the risk for type 2 diabetes. Conclusion. The journey of glucose from taste to cellular energy demonstrates the complexity of the body's signaling mechanisms. When disrupted, such as by artificial sweeteners, the body can become confused, leading to metabolic disorders. Understanding these processes highlights the importance of natural glucose regulation. At Mindful Diabetes Inc., through our Pathways to Wellness series, We aim to offer insights on maintaining healthy glucose metabolism by making mindful choices, especially in your diet. Avoiding artificial sweeteners and focusing on whole, natural foods can help keep your body in balance and prevent disruptions in insulin response.